My name is Ruben Sali, one of the sweet potato breeders at the International Potato Center based in Uganda. I'm also the product champion for boiled sweet potato. I am sharing with you the main highlights and project findings for the boiled sweet potato product during period 3. Let me take this opportunity to thank the team from various institutions that has contributed to this work during period 3. This presentation covers the progress we have made towards developing the boiled sweet potato product profile in Work Package 1, the biophysical characterization of quality traits in Work Package 2, the development of high-throughput phenotyping methods in Work Package 3, the integration of cooking quality traits in the breeding pipeline in Work Package 4, the on-farm evaluation of advanced clones in Work Package 5, and our perspectives for period 4. In period 3, we published the process of developing a food product profile for boiled and steamed sweet potato in Uganda in the special issue of the International Journal for Food Science and Technology. We highlighted gender differences in quality characteristic preferences. The improved variety Nasport 8 had the highest rating for overall liking in Kamwenje district and was equally ranked highly in Lira district. Sweetness and firmness were the key drivers for overall liking of boiled sweet potato. The raw roots desired for boiled sweet potato should be big with a smooth skin and hard, having a good smell and sweet taste. For proper processing, sweet potato for boiled or steamed product, the root should have a good taste, a good smell, and should be hard, easy to peel, and have a good appearance. The final boiled sweet potato product should have a sweet taste, good smell, be mealy and firm, and non-fibrous. In the longer list of desired quality characteristics, men and women cited different characteristics, with women often citing more characteristics. The penalty analysis of varieties used for consumer testing in Kamwenje district revealed that the overall ranking of varieties was penalized for not being firm enough, or not being sweet enough, or being too clear. However, the overall ranking of varieties was not penalized for not being mealy enough. The sensory mapping shows that Nasport 8 was liked very much by the consumers in Kamwenje district because it is sweet, smooth, attractive, yellow and orange, and is associated with the vitamins. While the variety Nderirabana was liked slightly because it is too soft, watery, tasteless, has no smell, sticky between fingers, and is not sweet enough. After translating the cooking quality characteristics of boiled sweet potato into lexicon in period 2, we have continued to train the sensory panel. Due to the safety guidelines due to COVID-19, SIP explored the possibility of conducting sensory evaluation from the home and office setting. The panelists were trained online using MS Teams. For home testing, each panelist prepared their own samples using a common kitchen SOP and evaluated the samples in their home. While for office testing, the samples were prepared in a central location and each panelist evaluated the sample in their office space. Home testing introduced variation due to cooking that was reflected in the poor panel performance, while office testing yielded good panel performance. The sensory profiling of the 6 varieties used in the office testing shows that the panel could distinguish the varieties by either the orange color and moist texture, or the mealy and firm texture, or the typical sweet potato flavor, or the fibrousness and soft texture. In the second season of period 3, we conducted sensory evaluation of the 16 parents of the Mwanga diversity panel, with NASPOT8 as a repeated genotype across the sessions, while simultaneously evaluating them for optimal cooking time and dry matter content. There were significant differences between the texture characteristics of NASPOT8 across the sessions, implying the heterogeneity of the texture characteristics of the boiled sweet potato. This means that to improve the performance of the panel across sessions, we need to have a standard genotype for various sessions. A moderate positive relationship was observed between sensory firmness and dry matter, which needs to be confirmed with a bigger dataset. We have continued to develop standard operating procedures for texture analysis. For instance, texture analysis was performed through a shearing test for standard cubes cooked at 85 degrees centigrade for 15 minutes with significant discriminative power. But unfortunately, this was not reproducible in successive harvests. To further improve our texture characterization, we have evaluated 3 sample preparation methods for texture analysis and made recommendations on how these methods can be improved, which will be tested in period 4. To understand the biophysical basis of processing and cooking ability of boiled sweet potato, a ring test was conducted between SIP, SEAT, JHI, and CIRAD. 16 sweet potato samples were analyzed for cooking time, firmness, and dry matter content at SIP Uganda, and milled, freeze-dried, and the samples were sent to SIP Peru for analysis for free sugars, starch, and proteins using NIRS. Another set was sent to JHI for B-amylase activity, PME activity, and cell wall characterization. Another set was sent for SEAT to be analyzed for free sugars by HPLC, starch, amylose, and RVA properties. Another set was sent to CIRAD to be analyzed for free sugars by HPLC, gelatinization, and amylose by DSC, and pectin content by calorimetry. Unfortunately, no correlation was observed between cooking time and firmness, cooking behavior and dry matter content, cooking behavior and starch characteristics, cooking behavior and beta-amylase and PME activities. These results cannot be conclusive because the standard operating procedure for cooking time was still under development. The ring test will be repeated with the optimized standard operating procedure for optimal cooking time in period 4. To develop NIRS calibration to predict quality traits, 3 to 4 roots of 60 sweet potato genotypes from the MGP population were scanned in different form by NIRS, either as raw fresh roots grated, raw freeze-dried and milled roots, raw fresh sweet potato roots horizontally cut, cooked fresh sweet potato cubes, cooked fresh sweet potato, and cooked freeze-dried and milled sweet potato samples. The NIRS spectra was used for calibration to predict various quality traits. For instance, dry matter content was predicted well from raw roots with or without grating and cooked fresh sweet potato samples. Fructose, glucose, and saccharose was also well predicted from raw freeze-dried and milled roots. Unexpectedly, starch and amylose content were not well predicted from most of the sample types. Cooking and firmness were also not yet well predicted. We shall continue with the development of calibrations for NIRS as a high-throughput phenotyping method during period 4. 25 sweet potato genotypes were used to sequence a 2.3 kilobase fragment, including the conserved region of the beta-amylase gene. A unique allele was discovered with SNPs and indels within exon 5 which forms part of the active site. This was used to cluster the genotypes into 3 groups. Group 1 constituted genotypes with low beta-amylase activity and beta-amylase expression. Significant correlation was observed between optimal cooking time and instrumental firmness, optimal cooking time and dry matter content, total starch content with dry matter content. Beta-amylase activity was negatively correlated with firmness. These initial findings provide a basis for further investigation, including validating SNPs in the beta-amylase gene as markers for firmness. For most of the work on phenotyping sweet potato for quality traits, we have used the Mwanga Diversity Panel, which consists of 16 parents and 1,800 genotypes. This population has been evaluated in the field extensively, and we are in the process of genotyping it as a training population for developing genomic selection models. Due to inconsistencies in establishment and reproduction, only a subset of 220 genotypes was planted in Namlonge, Serere, and Kabale during period 3. Almost 50% of the genotypes were able to establish consistently, and only 23% of the genotypes produced roots consistently. 27 traits and variables have been extracted from the sensory analysis standard operating procedures and have been included in the crop ontology. 13 variables for biophysical characterizations are under review as their standard operating procedures are under development. The sweet potato-based team is keeping track of the development of the crop ontology and making provisions of how those traits can be utilized. For instance, a user can now find a trait, create a list of traits, post-compose traits for new attributes, or request a new trait. During period 3, the sweet potato-based team also started making provisions for uploading NIRS data by creating provision for multiple analysis of a sample from the same plot, or making provision to upload NIRS data from multiple devices in a simple format. Using the standard operating procedures developed for optimal cooking time and texture analysis, we evaluated 8 genotypes selected for on-farm trials to be conducted by NACRI during period 4 under the DDBio project. During period 4, we shall continue to link the quality characteristics in the work package 1 to biophysical characterization in work package 2. Under the Sweet Gains project, we shall develop a comprehensive product profile for boiled sweet potato in the context of Mozambique. SIP will continue with the developing and validating standard operating procedure for texture analysis for boiled sweet potato. SIP will also continue refining the standard operating procedure for sensory analysis using the sensory panel for the RTBfoods at NAL, and train another sensory panel for boiled sweet potato in Mozambique with support from Sweet Gains project. We shall develop a new standard operating procedure for using texturometer such as penetrometry to predict mealiness of boiled sweet potato. The standard operating procedure for cell wall preparation, pectin analysis, and beta-amylase assay will be finalized. The cell wall fractions extracted by SIP funnel, JHI, and CERAD will be analyzed for uronic acid, monosaccharide composition, and methylation pattern to determine relationships between cell wall properties with cooking quality attributes. In work package 3, we shall continue with the NIRS calibration development for predicting cooking behavior and texture traits. We shall also optimize predicting texture attributes by processing images with the DGI. In work package 4, we shall validate the beta-amylase marker for firmness, screen our elite parent clones and advanced material for cooking qualities using our sensory panels, continue with consultations between sweet potato-based team with the sweet potato breeding team to harmonize sweet potato-based hide-up and crop ontology. evaluate the Mwanga Diversity Panel for genotype by environment interaction for quality attributes, while in Work Package 5, we shall conduct on-farm trials with NACRI with the DDBio project. Finally, we are grateful for all the support and cooperation from all our partners.